So VectorY is based in Amsterdam, the Netherlands. We're about 70 employees strong, founded in October 2020, so we just turned 2. I am privileged to work with a very experienced management team. Together we have over 100 years of experience in gene therapy, but also the production of viral vectors and other biologics, and a strong development background in big pharma and small and middle biotech. And together with this experience, we feel we are enabled to deliver on our mission, which is really to transform the lives of patients with serious neurodegenerative diseases. And today I want to tell you a bit about our programs, and afterwards a bit on our technologies which will enable these programs. Starting with ALS. We have 2 programs on ALS. And for those of you, I think everyone here knows how devastating ALS is. And in the United States alone, there's 30,000 patients with this really progressive and awful disease. And we are really passionate about curing this disease, making a transformational impact, because the medications on the market, they make a marginal difference to these patients. So before I tell you about our potential candidates, I first want to spend one slide on ALS itself. There's a really distinct difference between familial ALS, which is characterized by specific mutations in the patient, and sporadic ALS, which is around 92% of the ALS patients. Most of the drugs that have been tested in the clinic focus on familial ALS, so we really differentiate in the fact that we're going for sporadic ALS, which is the larger patient population. And with these sporadic ALS, we have a different toxicity. It's primarily because of lipid toxicity, which causes a secondary proteinopathy, usually TDP-43 aggregates. And in familial FALS, you see mutations that cause a primary proteinopathy. And it's shown in a recent publication where 4 microliters of CSF from either sporadic or familial ALS patients were administered to mice. And those with familial ALS did not develop any symptoms, while those treated with the CSF of sporadic ALS patients developed ALS symptoms and TDP-43 aggregates. So this is really a hallmark of TDP-43 symptoms, and it's all been characterized to be attributable to the lipid fraction. So what does VectorY do? We try to target these 2 distinct pathogenicities of sporadic ALS. We've got 2 shots on goal. I think it's important to say we with reason, go for 2 different candidates in the same indication. ALS has been a very difficult nut to crack, and I think it would be presumptuous to think only one alone would do it. So for the longer term, we might even think of combining these, but for now, we're developing them separately. So VTX1 really focuses on these toxic phospholipids that are in LDL particles, and VTX2 targets these intracellular TDP-43 aggregates. So I'll start with the first one. VTX1 really, we're standing on the shoulders of giants. Here at UCSD, Joe Whitson and Sam Tsimikas have spent decades studying toxic phospholipids, but more in the periphery. So on the left, you see that Joe Whitson and Sam Tsimikas identified these toxic phospholipids which have a different structure and form a neoepitope. And normal people like you and me, we develop antibodies against these neoepitopes. So they have identified this E06 antibody, which is actually a murine version, and they've shown that it can protect in different inflammatory models, in a NASH model, and they really show an erythrosclerosis, and they really show that neutralizing these toxic phospholipids can stop all of these progression of these diseases. So what we do at VectorY, we have taken this E06 antibody, we've made a single-chain variable fragment and optimized it, humanized it to make it compatible for human use, and what we do is we vectorize it. So we make vectorized antibodies, we call it VecTabs, and these antibodies are then expressed by the neuron after it's been transduced with our AAV. And what we've shown so far, it's all preclinical still, but we can protect ALS and also healthy motor neurons from OXPC-induced damage. That's the green you see down there. So that's, you can see that these are neurons stained with the green, and our E06 antibody protects the motor neurons from damage and neurite outgrowth. A control antibody isn't capable of doing this. On the right-hand side, what you see is first healthy motor neurons that are treated with a non-oxidized phospholipid. And then you see the transcriptome. We've looked at many, many genes, but highlighted our ALS genes. If healthy motor neurons are treated with oxidized phospholipids, you see a dysregulation of all ALS genes, characteristic of the disease. And we can rescue this phenotype when we intervene with our AAV expressing the humanized E06. So this is all still in vitro, and now we're working on recapitulating the experiment I showed before with the mice and the CSF of different ALS patients to see if we can really prevent these mice from developing ALS symptoms. We have already looked at biodistribution, and we do this in pigs, mini pigs. Instead of NHPs, they're like cats. They're 4 to 6 kilos. These mini pigs, even though they're not the big ones of 100 kilos, these are, sorry, I only do metric system. These mini pigs are 30 kilos, so they're much bigger than a macaque, than an NHP, and their spinal cords are much longer. So we think this is a better species to really look at biodistribution. So you can see, we see the biodistribution in 2 routes of administration in these mini pigs, and we really see that the intracisternal route of administration, which is about here, shows the best transduction profile. Both are pretty good, but we see better transduction of the cortex. So we're happy with this transduction profile, and we will be moving indeed to toxicity studies as well to ensure that these vectors are safe. So that's VTX1. Now shifting gears to VTX2. So VTX2, as I said, is also on ALS, but on the TDP43. So here, we're not developing an antibody that is secreted to bind oxidized phospholipids on the cellular membrane. Here, we're developing an antibody that binds intracellular toxic aggregates and clears them. Now, it's important that we also leave nuclear normal TDP43 alone. This is because normal TDP43 is an extremely important regulator of multiple downstream genes that are required for normal motor neuron function. So we're very careful about looking at an antibody or a VecTab that binds only the toxic forms. So we've actually generated a panel. We have a very strong antibody group that have generated a panel of different VecTabs which bind different parts of the TDP43 protein. You can see there are the different domains, and we've screened all of these different VecTabs. So these are the single chain variable fragments with different number codes, and the same thing, we vectorize it. So the AAV encodes for the DNA for these different VecTabs. And then we transduce motor neurons, and we have a look what happens with regard to different functionality and capability of degrading these toxic intracellular aggregates. Here we have some data. So I'm going to take you through it. We have transduced different U2OS cell lines which express a TDP43 mutant which aggregates in the cytoplasm. And what we've done, we've measured the degree of clearance of the aggregates with the different VecTabs, so all the different ones we showed before. So it's very interesting to see that one VecTab is not as effective as the other. And I want to point out VecTab 51 because it'll come back later. It's not doing anything. It's not clearing of the aggregates. But other VecTabs, and honestly, we don't exactly know why there's such a difference in efficacy, because in general, it's just single chain variable fragment and has an antibody binding part. So we're working on that, but we see that there's a very big difference in efficacy in clearing these aggregates. But we have been able to reduce these intracellular toxic aggregates. And I don't show it here because of time, but we've also been able to show that the nuclear TDP43 is kept intact. So for the TDP43 program, we're also now doing preclinical studies in TDP43 mice. So for both VTX1 and VTX2, we're working on solidifying our preclinical package so that we can take it into, hopefully, the clinic in 2024. This is our envisioned clinical study, and I think what I want to point out most, it's really aimed at sporadic ALS patients. So that's really a large part of the ALS patient population. Already after 6 months, we want to envision the crossover of the placebo arm into the treatment arm, so we hope to actually quite quickly see if there's any clinical endpoints that we can measure. So I spoke a lot about ALS. I have one slide on Huntington, because that's actually our third program. It's a bit newer, but just as devastating, and also an intracellular protein aggregation of the Huntington toxic protein. And I just have one slide showing that here too, we have developed different VecTabs that bind the Huntington toxic transgene, or not, sorry, the Huntington gene in these patients. And you can see that some VecTabs are very good at lowering this toxic Huntington. So here too, we're working on a mouse model that can show a Huntington disease mouse model, R62, to show that this VecTab can hopefully change the disease progression. So those were the programs, and now I'm going to shift gears for the last few minutes to the technologies, because however fantastic your programs are, the foundation is really the technologies that are made to enable these beautiful programs and projects. We have 4 technologies. I've introduced VecTabs already. So this is the antibody group that really makes a really good binder, a specific binder to the toxic aggregate. The Vectron is a new transformative degron. It's a tag that we add to the VecTab to improve targeted protein degradation. And I'll show you in a second a slide on that. We have a capsid project. So really, here we focus on making safe and efficacious capsids that can bind specific target cells. And lastly, manufacturability. I think this is really the Achilles heel of gene therapy. I think we've seen enough presentations here to show that we're requiring really high doses and this complex manufacturing. So we're really thinking here with the end in mind. We have a lot of industry experience, and we're scaling up to 1,000 liters. So I think with these larger disease indications, we need to think of this from the start. So the degron pathway. What we do is we add a tag or a zip code to our single-chain variable fragment, which directs the protein and the single-chain variable fragment, once they bind, to a specific degradation pathway within the cell. And we have 3 flavors, and within each of the flavors we have different, so for example, we have 20 UPS degrons, and we have, within each category, we have multiple degrons. And so the top directs your protein to the proteasome, the middle to the lysosome, and the last one to the autophagy-mediated degradation system. And here, if you remember VecTab 51 that didn't do anything, what we've done here is we've fused the same single-chain variable fragment 51 to a panel of different vectrons, so different degradation tags for different degradation pathways. And you can really see that we get significant reduction of these aggregates for a single-chain variable fragment that didn't work in the beginning. And here too, you see also different degrees of efficacy of protein degradation. So we're very proud of this. We get a lot of positive feedback that we're really able to clear toxic aggregates, and that we're probably that we are really able to do this selectively and intracellularly. So one slide on the capsid platform. In view of time, I'm not going to go really into deep details, but we have really chosen the design of our capsid very carefully. So looking at different quality attributes such as immunogenicity or lack of immunogenicity, toxicity, increased potency. So potency here is really intracellular, how it escapes the endosome and gets the nucleus, and also this manufacturability. We use the baculovirus production system, which is capable, it has been used up to 21,000 liters for vaccine purposes, and we think this is the way to go to really have GMP-compliant, large-scale clinical batch production. And we have to design our vectors that they are compatible with this system. Here's some data on our capsids versus AV9 made on HEC. We really see high infectivity in human iPSC-derived astrocytes, motor neurons, and skeletal myotubes, and we can enhance this with our vector engineering team. The yields we get are fantastic. We've actually been to our CDMO, and when they saw this, they're like, wow, your upstream yields before purification are already 50% full. That's really high. And we're getting up to 6 E12 VG per mil on our harvest. So we're happy with this, and this will help us pave the way for really manufacturability at large scale. I told you before, we're going up to 2,000 liters, and at this moment in-house we have run our 50-liter process, and those are the yields you saw before. We're really looking carefully at every step of the way, knowing it's scalable, knowing it's compliant with GMP, and really looking carefully that we can produce such large batches. And this is then my last slides with 15 seconds to go. I think I hope I convinced you that we are passionate about generating these innovative therapies and that we're doing our best to make a difference for patients with ALS and other proteinopathies in the CNS. If you have any questions, thank you. Please come ahead, and thank you very much.